So I want us to look at the photosystems, a continuation of what we were doing last time, whereby we talked about that the photosystems is a cluster of primary and the accessory pigments, and we said that these ones, they are used to absorb the light energy at different areas of the uh, visible light spectrum. And we said that the photosystems are made up of two parts, the reaction center and the light harvesting center. Now we said that every pigment usually absorbs light and captures that light from particular areas of the light spectrum, which now leads to more energy falling on the plant being used. Although I mentioned that not all the energy that is actually transformed to the plant material that comes from the light energy, or that comes from the sun. We'll be looking at this when we talk about the energy transfers, as I said before. Now, if we look at this diagram of mine, you will realize that we have the antenna pigments and the photosystems. Now, when you look at chlorophyll A, we have chlorophyll B and the carotenoids. But what you need to know is that also on this region here, we have the specialized chlorophyll A. Now, this chlorophyll A you are seeing here is the non-specialized chlorophyll A. This is chlorophyll B, the normal chlorophyll B, and this the carotenoids, which are made up of xanthophyll and the carotenes. Now, you realize on this reaction center here, we have specialized chlorophyll A, and that is where we have the P680 and the P700, that is photosystem 1 and the photosystem 2. The non-specialized chlorophyll A, the chlorophyll B, and the carotenoids, they trap the light energy from the sun that is at different wavelengths, and then they transfer that energy, that light energy, in form of electrons to the reaction center where we have specialized chlorophyll A. Now, you realize that the carotenoids, the chlorophyll B, and the non-specialized chlorophyll A, this one, which are we refer as the antenna pigments or the accessory pigments, their role is to harvest light, that is to trap light energy and transfer it to the reaction center. Now, I want us to look at the two types of photosystems now in detail, but although we'll do it more when we're talking about the cyclic and non-cyclic photophosphorylation. Now, we can say that we have two types of those photosystems. So we can say we have photosystem 1 and photosystem 2 generally, and the photosystem 1 we said is the P700, that is its reaction center contains the P700, while the photosystem 2, the reaction center contains the P680. Now, the reaction center of the P700 is majorly found in the intergranum lamellae. You remember in our previous classes, we said that the lamellae acts as a skeleton that holds the granum in position, that is the thylakoid discs, to prevent them from clumping together, and that one would reduce the amount of light that is being absorbed. So they act as a skeleton to hold the granum in position, that is the grana in position, so that they increase the rate of absorption of light energy to increase the rates of photosynthesis. Now the P700, that is photosystem 1, is involved in both cyclic and the non-cyclic photophosphorylation. But when you look at photosystem 2, which contains its reaction center contains the P680, this one is majorly found in the grana, and this one is involved only in the non-cyclic photophosphorylation. Now, what is this photophosphorylation that we are talking about here? The word photophosphorylation is made up of basically three words joined together. We have photo, we have phosphor, then we have relation. Now, relation is a process of addition. Phosphor is the element phosphorus, and the photo refers to light. Now, when we talk about photophosphorylation, it means it's the addition of a phosphorus group or a phosphate to an organic, or let's say to a nucleoside, let's say to ADP, in the presence of light energy to form ATP. That's what we refer as photophosphorylation. Actually, the formation of ATP from ADP and inorganic phosphate. Now, we have chromatography as a method that we will use to identify the various dyes or the various pigments that are found in the leaves. And this chromatography is a method that you have come across actually in chemistry, where you learn how to separate the various dyes depending on how soluble they are in the specific solvent that you are using and how much they stick to that adsorbent material that you are using, or let's say if you are using a filter paper. So the substances or the dyes in this case will be using photosynthetic pigments. They are soluble in that organic solvent to different degrees, and they stick to that adsorbent paper at different degrees, and therefore they are able to travel different distances depending on their solubility and stickiness. Now, this chromatography is a practical thing that you need to understand what it entails. Now you realize that it involves three stages. It involves the extraction of the photosynthetic pigments. Then two, it also involves the separation of the photosynthetic pigments. Now, then it also involves the identification of those photosynthetic pigments. So you separate those photosynthetic pigments after extraction, then you identify them. So I want us to start with the extraction process, which is commonly done using a mortar and a pestle. So we want to look at how that extraction is done. So the first thing is to collect the leaves, that is fresh green leaves. You collect fresh green leaves. Then using a mortar and a pestle, you grind them in a solvent. Let's say you can use ethanol or propanol. These two, ethanol and propanol, they are actually organic solvents, and because the chloroplast pigments are also organic, it means that the ethanol will dissolve propanol. Like dissolves like, meaning that polar for polar, non-polar for non-polar. So in this case, the ethanol or the propanol will be used as the solvent to dissolve those photosynthetic pigments. Now, we want to look at the separation of those photosynthetic pigments. Now, this one is done using the paper chromatography technique, and the paper that you are using, let's say if it's a filter paper or we refer it as the adsorbent material, you cut it to a given size. Like in this case, you are supposed to cut it to 2 centimeters to 15 centimeters in this case, whereby now you form something like a strip that you are seeing in this slide here, this one here, whereby you realize, for example, this distance from here to here is the one that is supposed to be 2 centimeters. Then this one along this end up to this, where after the baseline, now that is the length, which we should be now in this case 15 centimeters. Now then the next step after you have cut that one, then you draw a pencil line 2 centimeters away from the margin, and that is called the origin. Now if you come to this slide of ours, you realize that this is the baseline. You are supposed to draw it let's say 2 centimeters, depending on the choice that you take. In this case, you can draw it 2 centimeters or 5 centimeters from the baseline. Now you are going to draw it in pencil so that you avoid using ink, because ink contains also dyes or different colors which may interfere with this pigment that you are going to place here from the extraction or from the extract that you have obtained from the leaves. Now once you have drawn that the baseline 2 centimeters from the margin, you use a dropper and then you add that green extract that you have obtained from the leaves, that photosynthetic pigment. So you add it and you allow it to dry. You allow to dry. So that's why we are saying in this point that using a dropper, you add the green extract on the origin repeatedly, allowing drying between the spots until you get a concentrated pigment spot. Now you add the spot, you allow it to dry, you add again, you allow to dry the same spot till you get a concentrated pigment spot. Then you add 2 centimeter cubic of petroleum ether into a boiling tube. This is where you are going to place that filter paper inside, or that strip of yours that you had cut here, that is 2 centimeters by 15 centimeters. So you are going to place it into the petroleum ether. This is going to be our solvent now to dissolve that dye. Actually, when you dip this paper, this filter paper inside that, let's say the boiling tube, let's say at this point here, along this line here, let's say we have a line here just below the baseline. That is where that ether or that petroleum that is where it is going to reach at this point here. You don't allow it to reach up here where we have the baseline, because it will dissolve this dye that you have placed here and you have allowed it to dry. It may end up mixing with it, and then the whole of this paper may be just actually the pigments may spread on the whole of this paper, and that one may not allow the formation of a chromatogram of this type that you are seeing, for example, in this next page. Everything will be affected and you can't see these colors separated out clearly. Now, once you have allowed it to stand in the petroleum ether or solvent in a boiling tube, then you vertically suspend now the paper chromatography in that boiling tube, make sure that the green spot does not touch the ether. I think I've mentioned something on that. Now, then you leave the setup for 5 minutes to 24 hours. And this one now will allow that solvent to rise up the paper. Remember, at this slide we said that the solvent is going to reach at this point. Remember, this is an adsorbent paper or a filter paper. It will absorb the dye, and that dye will start moving upwards. Sorry, the solvent will start moving upwards. It will absorb the solvent, and the solvent will start moving upwards. When the solvent reaches at this point, it will actually dissolve the dye. And depending on the different solubilities of the dyes that are present here, that is the pigment, photosynthetic pigments, and the way they stick to this filter paper, then they will start separating out. Now, for example, this pigment you are seeing here, it's more sticky and less soluble. Meaning, those pigments that are more sticky and less soluble, they will travel a shorter distance. Those that are less sticky, like this one here, and more soluble, they will travel the longest distance along that filter paper. Now, then my third point here is that each pigment travels at different speeds. Hence, distance moved by each pigment is unique. They also have different thickness. Now you see, they are traveling at different different speeds because of the differences in thickness and solubility. Now the pigments are separated as they ascend, meaning is because of the different speeds they are traveling due to the different solubilities they have and thickness to the adsorbent material. When the solvent is nearest the other end, remove the paper chromatography and immediately draw a pencil line to indicate the solvent front. Now, if I take you to my diagram here, you will realize that this ether that you placed here, as it moves up, it will tend to move faster than the way these dyes are moving. Meaning, at some point, it will be ahead. Actually, in most cases, it will be ahead. In all the cases, not even most cases, in all the cases it will be ahead. But you monitor in that it does not go beyond this point, because if it's going to reach at this point and the dyes keep on moving and you allow it to stay for long, these dyes may move even out of the paper. So the moment it reaches close to this margin, you identify that point, you mark it, then you draw another line with a pencil. This one is to prevent other dyes, if you are using ink, other dyes within the ink from separating out and spreading, destroying or making this chromatogram of ours to look ugly or to look bad. Now, once you draw that pencil line, or once you draw what we refer as the solvent front, now you allow your paper to dry, and that one forms now what we refer as the chromatogram. Now, this level of the solvent front, the baseline, and the position of the dyes will be very important in calculating what we refer as the retardation factor, and that's why you should identify the level of the solvent. That is the farthest point that the solvent will travel in that particular experiment. Now, when the solvent is nearest as the other end, as we have said, you remove that paper chromatography and you draw a pencil line to indicate the solvent front. You let it dry and it's now called a chromatogram. Now, how do you identify the photosynthetic pigment using the retention factor, or what we refer as the retardation factor? Now, the retardation factor is usually a ratio between the distance that has been moved by the pigment to that has been moved by the solvent. Now, that's what I'm saying here, that the retardation factor or retention factor is the value, is the distance moved by the pigment from the origin to the center of the pigment, and this one is divided by the distance moved by the solvent. For example, if the dye X has traveled 10 centimeters and the solvent front is 17 centimeters, then you will take the dye X distance, that is 10, divided by 17, and that one will give you the retention or the retardation factor. Now, one thing I want you to note is that if person A in Kenya, or let's say Mary in Kenya, and let's say Amna in let's say USA, and let's say Abdraman in Pakistan, if they do the experiment of these photosynthetic pigments and they all use ethanol and different times, meaning their dyes will travel different distances. When they calculate the retardation factor, it will be the same for all of them because they have used the same solvent. Now, if you change the solvent and each individual uses a different solvent, then the retardation factor for each pigment will be different. That is the same pigment for all the three individuals. But if they use the same solvent, then the pigment, let's say if it's xanthophyll, phyophytin, let's say carotenes, or chlorophyll A or B, they will have the same retardation factor in all the three individuals because they have used the same solvent. But if they change, then they will have different retardation factors. Now, if you look in this diagram, we have said that, for example, here the first statement here, we have said that the retardation factor or value is given by the distance traveled by the solute or the photosynthetic pigment in this case over the distance that is traveled by the solvent. Now, this one is very important because, like, if you want to look at A here, is the distance that has been traveled by this pigment at this point, that's pigment capital A, has traveled distance small a, and then the solvent front has traveled distance x. So the retardation factor is given A over x. So A is the distance traveled by the solvent and A is the distance traveled by the dye A. And that now will end up giving, for example, at the end of the experiment, we'll get something like this. That is the carotene, phyophytin, xanthophyll, chlorophyll A, chlorophyll B, and this is the loading point where you placed your your photosynthetic pigments. Now, in our next class, we'll look at the absorption spectrum of photosynthetic pigments. We also look at the action spectrum of the photosynthetic pigments. Then we will further look at how are the two correlated, how are they related to one another, and how do they determine the rate of photosynthesis.